So I think we start with our CMA. So COVID has caused us great loss. It is not only physically, mentally, but it has taken our lives. You can see Dr. Sanjay Vadhavan was a COVID victim. But finally, vaccine is here. There are so many vaccines, so many companies manufacturing it. There are so many truths and so many myths and so many controversies which are associated with COVID vaccine. So to take you through a journey of COVID vaccine and about the immunization, we have this CME organized today. It is on COVID vaccine and update and where do we stand today. So to throw the light on the same, we have with us an eminent speaker, Dr. Professor Sonal Saxena, who is an MBBS from Maulana Azad Medical College and MD Microbiology from the same. She is presently Director, Professor and Head of Department of Microbiology, Maulana Azad Medical College. Presently, she is Administrative in Charge in the Department of Microbiology, MAMC, Nodal Officer, COVID-19 Laboratory, Lab Coordinator, Delhi's COVID-19 Zero Survey, a member of State Task Force on CBNAT and TrueNAT testing for COVID-19, Joint Secretary, Indian Association of Medical Microbiology. She has a special interest on antimicrobial susceptibility testing, practical implementation of technology for antimicrobial resistance, antimicrobial stewardship, bacteriology and infectious disease and medical education. She has WHO fellowships on antimicrobial resistance at ICHAR from Sweden from 10th February 2012 to 23rd March 2012. She has received a Best Poster Award on broad spectrum antibiotic short course use amongst children. She's delivered an oration, Shri Gridhali Lal Raturi oration at IMS Symposium at Dehradun. She is a WHO resource person to develop National Antimicrobial Stewardship Program for Government of Maldives, AMR training in Monap at Nepal and India. She is a member of various antimicrobial resistance committee and WHO task force. She has various research and publication and numerous students taking training and doing post-graduation under her. She had a big resume. So without wasting much time, I would request Dr. Sonal Saxena to please start deliberating her lecture. Dr. Sonal Saxena, please.
Thank you, Dr. Meenakshi. First of all, am I audible?
Yes, you are audible.
Yeah, thank you. First of all, I would like to thank your organization for giving this opportunity to share some of my experience and some of my thoughts about the topic. Now, Meenakshi, am I allowed to have to enable me?
To share your slides.
Yeah, you'll have to enable me.
Is it done?
Yes. So is my screen audible, visible?
The screen is visible. You are audible as well.
Thank you so much. So that's the talk of today. That where are we today and COVID-19 vaccine. Now we are all in a daze right now. We've all had a very eventful year, a very different year, a year which was full of tragedies, full of new things for all of us. But the year ended on a good note. And the year reminded us that where there is a will, there is a way. So this presentation is purely based on the data which is available in public domain, as I do not have any personal experience with the vaccine, except for the fact that we were involved in some trainings. So if we discuss the vaccine part, we all remember Edward Jenner and his very famous smallpox vaccines, and how we have moved from smallpox to COVID-19. And that gives us a very huge hope, because it tells us that if we could eradicate that, I think we will be able to eradicate a lot of things. So one good thing that emerges out of the whole thing is that this is going to go away very soon because we have a vaccine. But what are the attributes to it and what has the journey been like to develop this is also something which we all have to, see. And things is not moving. Yeah. If we look at the immune responses which come to a vaccine, it's something which all of us were very familiar with in our initial years of training in, MBBS, where we were taught that upon injection, the antigen presenting cells take up the antigen and then they present it to the T helper cells, and these are then processed and you get multiple responses. You may get a cell mediated response or you may get a humoral response where these cells become plasma cells and produce antibodies, which gives us one kind of immunity. And to remember that immunity, it goes into as a memory B cell or a T cell. Now, this is a very, very simplified version, but I felt that since this is a clinician platform, this would be the right way to remember that, yes, this is what we were looking for when the scientists went about designing a vaccine for COVID. This is what they had in mind. So now this is what happens in a normal vaccine. You know, there is an exploratory and a preclinical study which is done in animals with the antigens that we think are going to work. And this usually is a very long drawn process which takes about 2 to 3 years. Then, after we have identified the molecule which is likely to be immunogenic and is proven to be immunogenic in various animals, then it goes into a phase 1 clinical trial where there are just a few participants, usually about 10 to 20. Then it goes into a phase 2 trial where the participants are maybe around 80 to 100. And it goes into a phase 3 trial where the participants are in thousands. And it's only the result of these phase 3 trial along with phase 1, 2 that the company applies for approval and the approval is granted. So this whole process takes almost 7 to 8 years, and then the company starts manufacturing. And we have a post-marketing surveillance, or many studies would like to call it a phase 4 clinical trial, which goes on and is an ongoing process. And many vaccines have been withdrawn from the market. Many drugs have been withdrawn from the market. The biggest example being the Rota vaccine, which was withdrawn after the post-marketing surveillance. So that is also a very, very important aspect when we consider the vaccine development. So the question on everybody's mind is that if something takes 10 to 15 years, maybe you can shorten it to maybe 7 to 8 years, 5 to 8 years, but how can you shorten it to months? So the answer here lies in this, a very, very simplified version which was presented by these authors, where they said that since clinical trial, phase 1 clinical trial and the exploratory preclinical phases were merged initially, whenever they found a molecule, and the biggest help which the scientists got here was that on 11th of January itself, the Chinese authorities put up the whole genome of this virus on a public platform or a domain. Now, right now, genome is become the in thing, but the genomic studies take a lot of time. But now, with the new machines being available and the data processing being simple with such high CPUs, it has now become possible to sequence a RNA virus in, say, about 10 days or 11 days. And they were able to put up these, and that is what allowed the scientists to design those antigens and see the immunogenicity in a very fast manner. And at the same time, they started the phase 1 clinical trials, which are basically done to assess the risk factors, what doses are required, whether this dose is going to be different in different individuals, whether it promotes a healthy immune system to make antibodies, how will it work in those people who do not have a healthy immune system. And the phase 1 then got combined with phase 2. The phase 2 then got combined with phase 3. Now, phase 2 primarily aims at the safety and immunogenicity, and phase 3 tells you the efficacy. That is why the denominator is so huge, that the more you take, the more likely is a chance that you might not be able to screen out those people who may not be producing response. So if you take 100 and the prevalence of such cases who are not going to respond to this antigen is 1%, you will have only one. But if you take 1,000, then it becomes 10. And if you take 10,000, it becomes 30. So by a simple this thing, the efficacy studies of phase 3. Now, there is a lot of controversy regarding this. That is why I wanted to cover this part. And then the company applies for approval. What happens here is, in COVID-19, the companies have applied approval after phase 2. So companies have applied this approval after phase 2, and this phase 2 approval was be granted clinically only when there is a data which is made available to them after phase 3. So even though the paperwork was done after phase 2, the approval was not granted. It was granted only after evaluation of phase 3 clinical data. And before the approval was granted, if you look at this arrow, the phase 3, the manufacturing and the post-marketing surveillance will obviously not start, but the manufacturing started. That is why the moment even for India, the vaccines were granted approval. Within 10 days, the vaccine plan is being rolled out. Similarly, in UK and US also, they were able to roll out because they did not wait for approval and then went into consideration of producing these vaccines. So if you look at the various types of vaccines, we have a DNA, RNA vaccine or the nucleic acid vaccines. Till now, none of them were available. Now we have two RNA vaccines here. Then viral vector vaccines, Ebola and dengue, these were made available after 2005. The viral subunit vaccines, this we are all familiar with. The hepatitis B vaccine, even a rabies split vaccine is also available. For flu, a split vaccine is available depending on how it goes about. Then there are various viral-like particles which are made available for HPV. And then we have inactivated vaccines and the live attenuated vaccines, which we literally grew up with. The polio vaccine and the vaccines in the immunization programs, which are as such being used for ages. Now, what did we look at in COVID-19? COVID-19 is a very different virus. It uses what is called as a spike protein. Every virus needs something to latch on to the host cell, only then it is, otherwise it's a crystallizable form. So this uses what is called as a spike protein, which has got two parts. One is the attachment part, which attaches to the host cell, and the other is what fuses the two together, the fusion part. That is this is the fusion part, and this is the S1 part, which is the attachment part. Now, if you see this, it again has got a receptor binding domain, and it is the antibody to this receptor binding domain, which is called as a neutralizing antibody. So what we are looking for in an COVID-19 vaccine candidate was that something which could, if we could use only the spike proteins, it would give rise to the receptor binding protein domain, and that it will elicit the antibody response. Now, the problem here is what happens if this spike itself changes, and that is the scare of the UK strain, that possibly this spike is changed to such an extent that the antibody which is produced against this may not work. Now, again, we'll have to wait for 6 to 12 weeks to see the antibody response. This is a step which we can't hurry up. Now, what are the various vaccines in pipeline? There are plenty of them. What started with 319 molecules is now narrowed down to just a few. So in the current stages, if this is from 24th November, so it's not very late. In the exploratory stages, we had more than 200 molecules, and preclinical stage itself, we had 87 molecules. And then in phase 2, phase 3, so these 6 vaccines which are there in the phase 2 and 3 are likely to be licensed in the next, 1 month or so. And why there are multiple vaccines? Because there is a race to develop a vaccine. All the vaccine targets which are picked up basically have to stimulate a primary immune response. Now, this primary immune response, as I said, is an antibody which is against this receptor binding domain. Because if you block the receptor with the antibody, the virus spike protein will not attach to that receptor. So we are all looking for that. At the same time, there is a lot of controversy surrounding the cell-mediated immunity, because we know per se that once the virus goes inside, nothing kills it. But if you have a very good humoral immunity, the virus will not go inside. If the virus goes inside, then yes, cell-mediated immunity is the only way to stop it. But if the virus is stopped from going inside, and that is what is the aim of these vaccines. So each vaccine has got its own selected distinct advantages and disadvantages, but the development of these different vaccines have got some redundancy and some overlap. The other thing was why there was a multiple race was that whatever each scientist group thought in each country went on to become maybe something will be successful. So in case a vaccine is unsafe for humans or fails to protect, you will always have another molecule. And then the biggest question was that these vaccines will allow the global population to be immunized sooner. No country will go begging, asking for a vaccine if you have your own vaccine. The other question which normally rises in our body is, in this discussion is that why we are looking at 2 doses. So we are all, again, this is a diagram which comes from the primary textbooks of immunology, from biochemistry and microbiology, that initially the body generates an immune response in response to either an infection or in response to the first dose of vaccine. And if there is a subsequent dose, then this response is bigger and this response is faster. So the secondary immune response also creates an immune memory. So that is why it is important for us to give 2 doses, and that is why most of the vaccines are made in such a manner that you give 2 doses. The whole world is doing active vaccine trials, possibly except for Africa and for Australia and obviously Antarctica. Many of the countries, the vaccine trials are going on. What you will be more surprised with is Canada has not done any trial but has booked the maximum number of vaccine. If you look at the chart for vaccine booked by each country. Now, what are the various types of vaccines which are being developed for COVID-19? Now, till now we have a very clear idea, and that this spike is the one that we are looking for. The scientists knew right in the beginning, even after genome sharing, that it is the spike that which we are concerned about. So one word I would like to say, remind you about, what are the nucleic acid vaccines, because these are the vaccines that we are seeing for the first time being developed. So ye ab tak kahan thi? Why didn't we get them sooner? So if you remember, the DNA transcribes the RNA and then the RNA transcribes the protein. Now in DNA vaccine, you give the naked DNA, which will later on transcribe an RNA inside our body, which will later on produce a protein which is responsible for generating an immune response. So if you want to generate an an antibody to the spike protein, what you do is that you give the DNA or the gene which codes for that spike protein. By DNA, we mean the gene. Then the other thing which came about is the RNA vaccine. Now, if you give the RNA vaccine, it is instantly transcribed into a protein. So further, there is no as such. Now, the advantage of giving the RNA is that RNA doesn't go and attach to the host genome, because our genome is a DNA genome. So it immediately transcribes into a protein and the rest is similar. If you give a protein, obviously it gives you an immune response, but each has got its own advantages and each has got its own disadvantages. So if in the mRNA, it is placed in a lipase action molecule, because what happens is in the laboratory also we face this problem. RNA is a very, very easily degradable molecule. RNA and DNA require absolutely nothing to kill. It gets killed on its own. That is the reason why all VTMs are transported. Why you need a VTM is because of this. Then why VTMs are transported in dry ice is because of this, because our environmental conditions are so bad that the RNA is going to be lysed by the time it reaches the laboratory. Even in the laboratory, everything is stored at -80. For this reason, if you want to preserve the RNA, all our -80 refrigerators are full with samples, because ICMR has told us not to discard anything. So this RNA has to be coated with a lipid nanoparticle vehicle. So imagine an RNA like a small thread which is coated with a bead which consists of lipid molecules. Now, once administered, this RNA is taken up by the host cells, and these lipases will degrade these lipase vehicle which has been used, and then it would get translated into the spike proteins. Now, the two most famous, the most glamorous vaccines, as I would call them, are the Moderna and the Pfizer vaccines, which are working on with this. Now, they've got each have got its own advantage and disadvantage. The advantage is that it elicits both humoral as well as cell-mediated immunity, whereas, and it is non-infectious. That means it is very safe for immunocompromised people. It gives a long-term immunity. It has a prolonged expression of antigen, which results in a significant immunological memory. It's very easy to develop and produce and a large scale production. And in future, probably multiple diseases, the gene can be coded on same mRNA and it can then be given. And there is many a times there is a risk. Uh, there was something circulating on WhatsApp that you'll become it's made up of some kind of a chimpanzee gene and you will become an animal. I mean, ridiculous. So it does not integrate into the host genome, and the RNA strand in the vaccine is degraded once this protein is made. That's how our body works. Everything is made by mRNA is the template for protein. So the moment the protein works, the mRNA gets degraded. The disadvantages, actually we don't know how it's going to work because this is the first time this kind of vaccine is being produced. We believe that it's going to go and hit very hard to our immune system. So the immune response is going to be very, very high. But if the immune response is going to be very, very high, whatever goes earlier comes down earlier. So the response is going to come down also very soon. And the most important disadvantage is the storage and delivery. As I said, it's very, very difficult to preserve mRNA. If you imagine the country where uh, there is a district health center and then there is a sub center and then how many people are going to be vaccinated if you can't store this vaccine properly? If the cold chain breaks, the vaccine becomes useless. And vaccine creates a very good false sense of well-being. So you feel you've got a vaccine, now you're safe, and the vaccine never worked. So that risk is there with these vaccines. Still, Moderna and Pfizer are working very well, and in the cold weather, in the cold conditions, it's not that risky. But imagine the vaccine being administered in Cochin or Kanyakumari, the situation would be totally different. Again, the target antigen which is being used by the mRNA in both these vaccines is a spike protein. It's got 2 doses, 28 days apart, 4.5% efficacy given intramuscular. The side effects which have been reported are not much high. Both humoral and cellular responses have been detected, which peaked at day 15 after the first dose. That's very, very surprising because what happened after this? And there was one of the trial also said that half a dose worked. So that is one for issue for, I think it was Pfizer. Then Pfizer has had 4 or 5 adverse events. The rest of the things are all similar. It's given in 2 doses, 21 days apart. I think that is the British and the US differences, that they are giving 21 days apart, possibly just to differentiate it from the other. It has 4 serious adverse events have been reported till now, which were serious events in the trials. One was a shoulder injury, the other was a right axillary lymphadenopathy, paroxysmal ventricular arrhythmia, and right leg paresthesia. So these vaccine trials have been, right from the beginning, have been riddled with controversies, and their humoral response peaked at day 7 after the first dose. So this works really very fast. This is also approved. Then we have the DNA vaccines. As I said, the DNA vaccines you need to put into something and send again. So the best way to send any DNA in anything is using a carrier plasmid. Plasmid is basically a extra genetic material which is there in the bacteria. So the gene is attached to the plasmid and then it is delivered to the body. The plasmid is used because the body does not degrade the foreign gene before it can provoke an immune response. This plasmid comes from a bacteria and we are all familiar with that. So our body doesn't destroy this gene. So it gets transcribed to mRNA and then to protein. Now the vaccine here is the Inno 4800. The problem here is that it is limited to a protein immunogen only. Now this is in phase 1-2 trials. It also works on the spike protein. The phase 2 trials have found it to be 94% sync, both humoral and cellular responses, and it is 2 weeks, but it is given intradermal. Then the side effects, there are local side effects and there are some systemic side effects which are mild. This is in phase 2. Then the various vector vaccines. Now, vector vaccines are actually our friends. Vector vaccines means that the viral vaccines are you use a harmless virus, something which doesn't do anything in our body. So you can take an adeno, like you've taken an adenovirus from another animal and use it as a carrier, or you use a virus which is an attenuated virus, which you know will go and help the gene but will not cause a disease by itself. So this is the virus, when it infects the cell, it administers this foreign gene into the cell, because just like coronavirus, this virus also will use a particular receptor and will enter the cell. Once it enters the cell, it will deliver that gene inside the cell, and the whole story will begin. So there we were giving a naked mRNA. Here we are giving the whole virus. The advantages are that it elicits really a strong antibody and T cell responses. Like it's like a dry run for the system. It primes the system very, very well. Then it allows both B and the T cell to be activated because it's actually using a proper virus. Then the vector vaccines can be used in immunocompromised individuals. They cannot be used into an immunocompromised and immunosuppressed individuals. So that is one disadvantage, because you don't want to take a risk that in immunosuppressed individuals, maybe this vector can suddenly become a pathogen. Then it is less effective in people who have the pre-existing antibodies. So adenovirus causes common cold, diarrhea. So if you've had this infection with viral diarrhea or viral cold is so common that we all have antibodies to these. So the pre-existing antibodies can actually destroy this vector before it reaches inside, and they are a little complicated to produce, and they require specialized facilities because it has to be very, very pure. So then it needs a lot of R&D behind it. But this is the vaccine that we are getting. It is the Oxford AstraZeneca, which has been approved in India, you all know, on 1st of January. 2 doses, 28 days apart. Intramuscular efficacy is a little low, 70%. And how is it going to pan about in post-marketing trials? But it is one of the most sturdy vaccines. It doesn't require much storage. It has reported one case of hemolytic anemia, one case of transverse myelitis, and one case of high fever. And it is this transverse myelitis which is actually, given it a lot of bad blood. Then the other vaccines which are available are the famous Sputnik vaccine, which is from Russia. In India, it is under phase 3 trial, but it has been approved in Russia. Johnson & Johnson Janssen trial, it is in phase 3. They actually had to stop in between. Then CanSio adeno5 nCoV virus phase 3, it's a Chinese vaccine, and so is a one-tizimine, which is in phase 2, again a Chinese vaccine. Now, the live attenuated vaccine, we have all had these studies, and possibly the live attenuated, the virus is alive, but it has been rendered less effective. Now, why we did not have a live attenuated vaccine right in the beginning was that the attenuation takes time in laboratory. It takes serial passages, so you have to give that much of time for the virus to reach such a stage where the infectivity is there, but the virulence is not there. It is able to elicit an immune response, but it is not able to cause disease. So then we came to inactivated vaccines, where it was the easiest vaccine to produce. These have a whole pathogen which is killed or inactivated by chemical heat or radiation, and possibly eliminates the pathogen replicating and causing an infection, and has all the components that are likely to be encountered in the body. It's actually like a dress rehearsal for the body when it comes to immunity. The Sinovac, Sinopharm, and the Bharat Biotech Covaxins are the major stars for this. The problem here is that they are multiple boosters may be required, because to maintain the immune status in an individual. So you may require more than one booster. It is a little less effective than generating CMI, and it causes a considerable risk if the inactivation procedure is not efficient. Now, the all very famous, or the within bracket ill-famous Bharat Biotech vaccine, which is co-produced by Bharat Biotech, but the R&D is by ICMR. And it's a whole virus, 2 doses, 28 days apart, intramuscular, where both humoral and CMI, but the humoral is far higher than CMI. The high level of neutralizing, which remained elevated at 3 months. Now, no other study has shown this, that they have remained elevated at the day 104 after the second dose, and this trial is still recruiting. Then local and systemic side effects were predominantly mild. Pain was the only, the most common adverse side effects. Now, this is approved in a trial board, a very, very first time for India, and that is why there is a huge lot of. The other vaccines which are approved are the Sinovac vaccine. We've all seen those product inserts and those boxes which have been circulating on the WhatsApp for quite some time, where the date of manufacturing is from May or June. That was a Sinovac vaccine or a Sinopharm vaccine, which is made by Wuhan Institute of Biological Products and Beijing Institute of Biological Products, where they showed that by day 14, 75% patients had seroconverted, while the remaining seroconverted at day 28. So it's two dosage, where it is, they tried two, one 14 and 28, and it's given intramuscularly. So these are in use in China for past 3-4 months. So this is the only experience that we have, that this vaccine is actually working, the inactivated vaccine. The viral subunit vaccines take part in a pathogen which stimulate an antibody response. Now we've done everything. We've taken the virus, we've made it less virulent. We've taken a portion of the nucleic acid. We've taken the whole virus and used it. And the other thing is now that we use a part. So instead of using the mRNA or the DNA, we are using, say, only the spike. So we just use the protein or the particular subunit and inject it. Now here comes the Epivac Corona, which is already cleared phase 3 and has applied for approval in Russia. The Novavax probably has applied or is applying. Then by SCB-2019, which is still being developed. So Epivac Corona has got approval in Russia. The disadvantages are that it does not retain the native conformation. So the antibody, because it's a synthetic subunit, so there may be some partial activity, some impartial, some kind of poor antigenicity might be there. Whenever we look at the vaccine, we need to see that it's able to elicit a very strong immune response. So the strong immune response may be there, but you have to identify that subunit which it goes on. And antibody production, they produce a very strong this thing, but how long and as this maintains is not seen. The advantages are that obviously the strong antibody response, they're safe to administer and are simple and cheaper to make as compared to mRNA, and the delivery is also simple. Now everything goes into nano these days. The diagnostic chips have reduced to nano. The RNA detection has now come down to one cartridge based system, which is smaller than our microwaves. So everything is becoming smaller. So we also have a nanoparticles which have a natural lipid bilayer, and we also have an antigenic protein on it, and we have a non-infectious genetic machinery. The purpose of this is to have a structural protein which is co-produced. Now this is still in preclinical stage. It has not yet reached the trial stage. The purpose here is to have a natural lipid bilayer. Why do we have this lipid bilayer is because all our cells are made up of a lipid bilayer. So it recognizes it as fortunately or unfortunately, all living beings on this earth have got a lipid bilayer cell membrane. Even for bacteria and virus, that doesn't differ. So here it is able to recognize this very well and attaches itself. So these are still in the clinical stages. Now what has happened in India is, now this document is from the vaccine document operational guidelines, which was produced or released by MOHFW on 28th of December 2020. Till that, there was no vaccine which was licensed. So we can say that now we have a change of 2 vaccines. So Covishield or a chimpanzee adenovirus is by the All Lamarus Serum Institute of India, collaborator is AstraZeneca. This is now approved, and the Covaxin is now approved in a phase 3 advanced trial stage. Then we have a DNA vaccine by Cadila, Sputnik vaccine, which is in phase 3, which is to start by Reddy's lab, and it's recruiting phase 2 right now. The protein subunit vaccine again by Serum Institute, the Novavax. Recombinant protein antigen based vaccines by Biologicals Hyderabad, and mRNA vaccines from Genova, Pune by HDT. This is only in preclinical stages. Similarly, inactivated rabies vector platform with Bharat Biotech, again in preclinical stages, and a vesicular vaccine. This is a vesicular vax platform, meaning that it is basically a vector vaccine which is using a vaccinia virus, which is of animals. Now, what are the future challenges? As we set on, the biggest challenge is that are we going to uh, inoculate the whole globe? So much, the picture sounds so much simpler, but it actually conveys the whole message of the challenge that we are going to face in the coming 3 or 4 months. The whole world is going to face this challenge, particularly the developing countries. The other is that somewhere or the other, the whole process has been compromised. Everything has been, fast forward में चला दिया है सब कुछ. So there is a concern of an unknown side effects which can appear in a larger population. As I said, if a side effect appears in 0.1% of the population, unless you recruit 1,000 people, you will not have a single case of that vaccine. So if a trial recruits 10,000 people and it says that they had a one case of transverse mellitus, the concern that it raises when you replicate it to crores becomes very huge. The platforms which are based on nucleic acids, till now we do not have a successful vaccine, so we don't know how it's going to pan out. Then mRNA vaccines use lipid nanoparticles which are extremely temperature sensitive and may pose difficulties when you're scaling up the production, storing it, and after March, even the coldest area of the country will become, uh, you know, useless for this kind of vaccine. For DNA vaccines, reliance is on an electroporation or an injection delivery system for vaccine administration. Just like if you remember the smallpox vaccines, there used to be a tiny needle which used to go. Similarly, we will need an electroporation of an injector delivery which delivers it right inside the cells. So that needs training, that needs concerns. Then pre-existing immunity to adenoviruses is a concern, especially in a country like India, where these viral infections are very, very common, and our antibodies are always, updriven. Then rapid large-scale manufacturing have remained a challenge, even for polio, MMR. We've always had our challenges. Then mutations of the virus can result in vaccines which can lower down the efficacy. So this we are already scared about. Why UK has gone into a lockdown is because of this. That probably the vaccine is, and we will not know till another 6 or 8 weeks pass. We will not know about the vaccine as to what immune response effect it's having on the immune response, and if there is a vaccine enhanced disease for inactivated vaccine candidates. Now, a very, very glorious plan which India has made for the COVID 19 vaccine operational guidelines. And one should be really amazed. We run the world's largest immunization program because by sheer numbers, we are too many. So if you look at this document, it's freely accessible on Ministry of Health website. It talks about a multi-level governance mechanism, which has been laid down at every level. That on national level, you will do this, on state level, you will do this, on district level, you will do this, on the sub-district. These are the people who are going to be involved, how the DM is going to work, how the IDSP or the district surveillance officer is going to work, and how these sectors are going to converge together regarding the transport, the delivery, the injections. Then human resources training and capacity. We have been doing the trainings for about a month now. The I can talk about Delhi. Delhi has had about 30, 40 training centers. There is a training going on in Sachivalaya. There is one going on in my college on the third floor. So there has been a massive training and capacity building for this. And one most important thing is the intelligent network, that the digital platform which has been created, which is going to monitor as that the vaccines do not go haywire. They are not given to people who are not registered for it. Then the administrative, and then the logistics, cold chain management, communication, and social mobilization. We all live in a big city. We all feel that, oh, mobilization kya karna hai? Announce kar do, log aa jayenge. But people have to travel. They have to take a vehicle. They have to come on the day they have been told to come. Then who's going to monitor the adverse events? What happens? How it happens? As I said, post-marketing trials have been detrimental to many vaccines. So how is going to work out with this? We do not know. and the whole thing has to be monitored and supervised. So all this, and in our vaccine plan, the initial phase 1 is going to work out for the workers. So the phase 1 delivery is going to work till April, and there are 1 crore healthcare workers which are there for first, then 2 crore frontline workers, and more than 27 crore priority group, which right now is 50 years with comorbidity. And a few days ago, we were having a meeting, VC, with where Dr. Guleria said that they're going to identify these comorbidities as well. And they have a state task force meeting for that, where they're going to work around and even identify and define what comorbidity is going to fit into this. So this is something which is from America. This is from CDC. Sorry, the reference is missing. So CDC also had a similar thing where they started with healthcare personnel and nursing home residents, because their initial outbreaks in nursing home residents were very poor. So this is the status of the approved vaccine in the world. The inactivated vaccine, the Sinopharm and the Sinovac, were the first few vaccines which were licensed by Sinovac, and then the BNT vaccine, which is the Pfizer vaccine. Then we have the various peptide vaccines, or the Epivac Corona, which I said has been approved in Russia recently. This is from, if you look at the source here, this is a vaccine tracker. So if you Google vaccine tracker, or if you write this raps.org, it takes you to a live vaccine tracker, which tells you which vaccine is now in which state, in which country, which vaccine has asked for how many vaccine doses, everything it tells you. Now, these are the few references, and I would like to acknowledge here the help in this topic from my postgraduate student, Dr. Sanchita, who really did a lot of work for me for this PPT. So thank you so much. So the question on everybody's mind is this cover page of India Today, which came last month. Is it safe? Will it work? And if you can answer these 2 questions, I think everything gets answered for us right now. So thank you so much for this opportunity. Any questions?
Thank you, Dr. Sonal, for such a comprehensive, elaborate, and A to Z coverage of vaccine, starting from, I think, physiology, microbiology, and the clinical aspect and the execution. I think you have covered it brilliantly.
Thank you.
And it's
I
really,
would say that Z was missing. Now you miss the Zydus vaccine. In the DNA vaccines, I was hoping some data on Zydus vaccine. So let us say that it is A to Y.
No, sir, Zydus vaccine I have covered in the last page, where I've said that it is under trial. It is still not approved as yet. So unless we approve DNA vaccines, I've covered. So it is a DNA vaccine, but it is under trial right now. So
I expected, say, try a slide like Inovio vaccine, which is another DNA vaccine. So I expected something more on it.
what was there?
So Dr. Sonal, there has been a lot of controversy about in the newspaper about giving early authorization to this Covaxin. So what is your take on it? Is it all political, or should we be worried about the early rushed authorization of the vaccine?
See, I don't know what kind of controversy is there for that. It's an inactivated vaccine. Similar platform vaccines are working so well for past 6 months in China. The inactivated vaccine platform is the only vaccine which we have data of post-marketing surveillance. They are in phase 4 in China. Only why country insists on a data from their own country is because our ethnicity, our genetic makeup is slightly different from other country. So every country, whether it is FDA, whether it is CDSCO, they want their own country's data in phase 3. They have recruited 22,500 already in their trial, and they have reported very few adverse events till now. They have been recruiting since December. They are recruiting in one of the most prestigious organizations like AIIMS Delhi, Sir Gangaram Hospital. So these places, I think they are in the process. And in a clinical trial mode, they have been given is just because there is a safety net mechanism for each regulator. In a trial mode, there is some kind of a fallback for the regulator that I had approved it in a trial mode. But if you ask my personal opinion, if given a choice, I will take Covaxin.
Fully agree with you. I also, because it is a much more safer vaccine. There have been
We
instances.
know that.
There are only two vaccines which have been approved in India. One is the Covishield and another is the Covaxin. If given a choice, I would like to go for a safer vaccine because this is the only vaccine in India which is safest. The incidence of side effect is really zero. See, and we are going to give this vaccine, as the madam said, to the non-infected people who are absolutely healthy people. Even if one person dies, we have negated the effect of vaccine. Say if taking the mortality to 1.5%, we have negated the effect of vaccine, giving it to nearly 150 people, which is a huge number. And Covaxin is the only vaccine which has not shown any side effects. And the Covishield vaccine has shown 3 cases of, the madam is very polite saying that it was 1 case. It has shown 3 cases, and it is very difficult to say that 3 cases they did not happen because of the vaccine per se. Any person, any physician who has seen a patient of transverse myelitis can easily say that it is a debilitating disease carrying over for a very long time. And you give a vaccine to somebody who is healthy and you make him paraplegic, it is a disaster. The vaccine should be working on a zero harm basis. And the phase 1 data which has been published, though it is still not peer reviewed, it shows excellent immunogenicity and excellent safety.
Are
Right.
there 3 cases of transverse myelitis or 2 cases of transverse myelitis?
Actually, if you look at the literature review, Meenakshi, it says that the other 2 cases were not linked to the vaccine. They have very clearly specified that there were 3 cases of transverse myelitis, but only one they could link to actually to vaccine. So that is why the trial was also stopped in between. That because the trial gets stopped till it is evaluated that it is because of what. So they say that the reason is because something else and not the vaccine per se.
Let me elaborate it further. There were 3 cases of transverse myelitis reported in the entire phase of the Covishield trial. One case, they basically tracked it down to the undiagnosed multiple sclerosis. And one case belonged to the placebo group. So they say that it is not the placebo one itself was the meningococcal vaccine. And third case, they could not really deny. I really doubt the credentials of all those people who are involved into saying that it is not because of a vaccine per se when a vaccine has been given, because there's a lot of money on stake. Large number of people have been working, and it is not very easy for them to accept that it is not it is vaccine related. It is going to it is not going to kill all their hard work.
And the other thing is that multiply that 1 case by 30 crore.
That's what exactly.
If you increase the denominator to 30 crore, 30 crore is the phase 1. So 1 is out of how many? 5,000. So out of 30 crore, what is going to happen? So that is the worry right now. But and the other thing is that there were many reports earlier in media saying that a person becomes positive. The stage for that is that we have to remember all these studies which I covered, all the trials very clearly say that it takes 15 to 28 days. For Covishield, about 28 days for neutralizing antibodies to appear. And for the rest, the lowest is 15 days. That is with Sinovac. So if it's going to take 15 to 28 days, that is the time where you're most vulnerable and most relaxed because you feel you've got a vaccine. So the other is that we have never, on the whole earth, we've never attempted adult vaccination to such a magnitude. That is another story. We vaccinated the children. The children are brought to the immunization clinic by the parents. So the moment a vaccine comes, the anti-vaccine comes along with it. कि इसके साथ तो ये है और उसके साथ तो ये है. So if you have the kids, you can bring. But what will happen to an adult, especially with these news of transverse myelitis? So that's why I said we are embarking on the biggest challenges of our life. And like we've seen 2020 teach us new things, I think this vaccine delivery program is going to teach us more things about this portion also.
Madam, may I request you to kindly elaborate something more upon the hemolytic anemia that occurred with the Covishield vaccine?
Sir, I just read this much that there is one case of hemolytic anemia which occurred, and what they have done after that is not, and they've only marketed it in the trial. They've mentioned it in the trial that this occurred. The reason for that, even I could not find, because if something is going with a vector vaccine, it should not have a direct relationship with causing an hemolytic anemia. It can cause maybe a bone marrow suppression, but it cannot cause a hemolytic anemia.
Yes, Dr. Shukla. Unmute yourself. We can't hear you, Dr. Shukla.
Hello.
It's okay.
Can you hear me?
Yeah, you are audible.
Yeah. Thank you, Sonal, for such a nice lecture.
Thank you,
And
sir. Namaskar.
Namaskar. So I was listening throughout, and it was very nice. In fact, it was pleasurable to listen to you. Achha, one question I wanted to ask. I have become COVID positive. So should I go ahead with the vaccine?
Yes, sir. Yes, sir. You should go ahead with the COVID vaccine. The reason for that, this is one of the FAQs which Dr. Guleria answered day before yesterday in his VC. And the reason for that is that the immune system that the vaccine triggers might be a little different from an untrained immunity which we get when we get a vaccine, when we get an infection. There are very few cases of reinfection reported, but why should we take a risk? And we've already seen how its genome is changing. So in a span of 1 year, it has changed so many times. So it's better to take a vaccine even if you've become positive.
I think there is a consensus that those people who have had a mild disease, they should not refrain from taking the vaccine, because the virus, the nature of the virus is such that it is going to suppress the immune system. And the suppressed immune system develops how much of the immunity is yet to be seen. And while the vaccine is one thing which has a what we call as adjuvant to it, which boosts the immunity. So those people who had a mild COVID should definitely go for a vaccine. Which vaccine? That can be an issue.
No, only one thing I wanted to know, that because our antibodies has crossed 160, 170 like that. So in that case, should we go ahead with further vaccination?
Sir, it's only act to as a booster only. If not anything else, it's a booster for you.
Yes, exactly. That vaccine is going to act as a booster.
Okay, that's fine. That's fine.
I would like to dwell on one thing, which is the efficacy of a vaccine. Madam, highlighted the fact that the Covishield in itself is around 60%, 70% effective. Right now, the key question is not that one should be looking for is the effectivity or the efficacy of the vaccine. The prime importance for an individual like all of us should be the safety of the vaccine. All of you remember that the coronavirus is known to have a R0 factor of say 2.5 to 3. Though in my literature review, I have not come across the times when the coronavirus R0 was more than 1.7. But even if we presume from the various data which is available on the net that the coronavirus R0 is 2.5, even then we are bound to vaccinate about 60% of the population to develop a good herd immunity. So like just to be running after this vaccine, RNA vaccine from the Pfizer, that with a 90% efficacy makes no sense. Because as the other Poonawala says, that this vaccine might be 90% effective, but it may not be available to the 90% of the people in the world because of its limited manufacturing capability by the Pfizer and difficulty in producing this vaccine. So one should not go for the efficacy of the vaccine. Rather, one should go with the safety of the vaccine. The RNA vaccine, the Pfizer vaccine, is likely to be available in India. They have already applied for the license or the approval, though it may be available to the few privileged people on cost, and the government may not give it to the masses. But the vaccine has developed a good number of reactions. There have been instances of anaphylaxis in the UK. There are about more than 5 people have had severe anaphylaxis where the attendant drip was required. In India, the vaccine is likely to be given in the primary health centers, even in Delhi, the Delhi dispensaries. They are not, I don't think they are equipped with the personnel or the equipment or the medication to deal with such severe emergencies of anaphylaxis. So, in my opinion, my friend in itself from UK, he told me that he had a Pfizer vaccine, and he was down with so many complications for nearly 4 or 5 days. And many of our colleagues have been asking us which vaccine they should go for, and whether it is the Covishield vaccine or if they are given a choice, they should go for the Pfizer vaccine. As of now, my opinion goes with Dr. Sonal, that if given a choice, then I would love to go for the Covishield vaccine, for the Covaxin, that is the Bharat Biotech vaccine, because of its excellent safety profile. We have been going through the corona pandemic for last 10 months, and God has been able to save us. What if we do not get a good vaccine, but we should not have a bad vaccine?
I agree, sir, because like they say, a known enemy is better than an unknown friend. So at least the concept of mRNA vaccine, the biggest, as a microbiologist, the biggest thing that I feel is our immune system has not been exposed to something like this. And the allergic and the anaphylactic reactions are I think is because of the lipid molecules which are being used, or some protein molecule which is being used, which is getting the system into an overdrive. And the delivery of the vaccine is going to be a huge task. You're talking about anaphylaxis. When we started our RNA testing laboratory, we have done up to 600 in a day. We did not have a -80. We had to fight with the Delhi government to give us a -80 refrigerator, because it was a requirement of ICMR that we cannot start the lab. So then only it was bought and given to us, because we said we cannot start it. So if the country's #2 medical college did not have a -80, you can imagine what would be the situation be in an utmost periphery. So a DNA and RNA vaccine are very, very difficult to monitor their cold chain and to make them available at, maybe in metros they may be available, but in the periphery it will be very, very difficult for us to manage that.
Dr. Sonal, I wanted to know whether in one of your slides you said in the mRNA vaccines they have a prolonged expression of the antigen. How long, how prolonged is the expression, and like what makes you say so?
See, prolonged expression means that it's not that it's, see, mRNA, once it goes inside, it cannot make a copy of itself. It just produces and trains the system to produce its protein. So by prolonged, we mean it's longer. It's in comparison that it is longer than the protein, because the protein which you give in a subunit vaccine or in a virus vector vaccine, that can be degraded. वो गया और वो degrade हो गया body से. This is because it is going to produce, it will produce to a such an extent that it doesn't get degraded. So that is what we call as a prolonged this thing. And it works. The body is not able to call it foreign. So it doesn't, degrades it that faster. It's like any other mRNA which is producing the protein for the body.
As far as I know, that the mRNA is a transient phenomena, is a temporary thing.
It will produce the protein and go away.
That's what.
It will be degraded by the RNases. That is a problem even with the testing also, that the RNA gets degraded even in our laboratories. If we use an RNA solution too high, it gets degraded by the system itself.
Okay, so now what should be the ideal difference between the two dosages?
As the manufacturer says, 21 to 28. I think Covishield is 28 and vaccine is also 28.
So with our present program, we'll be able to manage that particular right difference. Like UK is, they are coming up with the thing that they are delaying the second dose because of number of patients increasing, and they are stressing more on the first dosage to whatever effect they get. So we'll be able to manage in India the two dosages at the right time.
We will have
the present.
I think with the COVID digital platform, it's going to send reminders, and it's going to send them. It's going to. What I learned from that document was that once you are registered with that, that is why all the phone numbers have been asked, all the healthcare worker phone numbers have been asked. Once you are registered on that CoWIN platform, they will give you a time and a date for your vaccine. And if you miss that, then you go back in the line, but not for the booster dose. The booster dose, you have to come on that day and take it. That is what the platform is going to remind you.
Platform is.
The
No, like, how can you
Will,
take a booster
will the
dose?
doctors have a choice to take which vaccine?
I don't think so.
So whatever vaccine will be available in that particular area,
Yes.
that is what will be given to us. And both the vaccines are being distributed across the country.
As far as I know, January 13th is the day they are going to roll out the program. So I think only then we will know which vaccine has been approached, has reached this delivery platforms. We can't say before that. Or maybe somebody will release a purchase order. किसको गया है तो आ जाएगा. Because Government of India must have released a purchase order for vaccine to either Serum Institute or Bharat Biotech. So वो कहीं ना कहीं से आ जाएगा तो पता चल जाएगा कौन सी आ रही है.
I think it is going to be Covishield, which is going to come first. I don't know in case Covaxin can also come. But I'm really concerned in case I have a call and I'm not able to go on that day because of certain contingencies. What happens to me? And in case I want to refuse, when do I get the next term?
As far as I know, the FAQ in the PT, they clearly said that if you miss your first dose, then you miss your chance. You will be put in the last in the line. So maybe they devised some mechanism for it. I don't know.
Regarding the question which was put by Dr. Meenakshi regarding the dose schedule, if we go through the trial which has been published in the Lancet, they have tried all the sorts of dose schedule. The trial was basically designed for a one dose schedule. Later on, they changed the code and they made it to the 2-dose schedule, right. 0 and 28 days, or maybe 6 weeks, less than 6 weeks. They basically broadly want to say less than 6 weeks. And some of their recruitees, they also got the vaccine more than 6 weeks, that is up to 12 weeks. The median was around 10 weeks. So they say that the vaccine has been working fine even with the 28-day schedule and even with a 12-week schedule. And the efficacy is nearly the same in both the schedules. And that's the reason that the Joint Vaccination Committee of UK has said that it is important right now to cover the large population, because even their single dose vaccination program has shown efficacy of say 63% or so. So that's the reason that they have spread it to take it to the larger number of population, large number of people. They have spaced their program to 0 and 12 weeks schedule. And they are taking recourse into the vaccination data which was available from the Covishield trial.
Sir, actually for every vaccine, more than the vaccine, it is the compliance which becomes an issue. So if you delay the booster, say by 3 months or 7 months or 6 months, all of us have been vaccinated with hepatitis B. Did you remember to take your last dose? Most of us forgot. Had the sister not reminded us or had the card not been there, we would have forgotten the 6 months dose. So the longer you delay, the compliance reduces. That is also an issue. तो अगर आप उसको 6 हफ्ते 8 हफ्ते 10 हफ्ते करेंगे वो आएगा ही नहीं लौट के. Same thing which happens with why you switched from erythromycin to azithromycin, because once a day compliance is always better than twice or thrice a day. So similarly here also, although one dose, as I showed you the primary and secondary immune response, it's always better to give a booster so that the immune system gets boosted. Plus the data that we have for COVID-19 is very less. We actually don't know how long these antibodies last and how, how frequently they need to be boosted. So we are all walking on an untrained territory and learning from each and every step.
Madam, another thing that our few colleagues would like to know, is it possible to take the primary dose of some brand of the vaccine? Not the brand, say some make of the vaccine, say the Vector vaccine, and can we take the booster of the different platform? Because all these vaccines, they basically stimulate the production of the spike proteins.
Sir, spike protein has got various epitopes. Epitope is the antigen site which elicits the antibody production. अगर दोनों वैक्सीन का एपिटोप अलग हुआ तो दोनों अपना अपना प्राइमरी रिस्पांस बनाएंगे और वो मर्ज नहीं करेगा. So and plus the vaccine, if the principle is different, then also it will not So it's better to stick to one.
That's really the thing. Do you feel that the Covaxin is going to have the immunity against, because there will be so many epitopes in the whole virus inactivated vaccine? Do you feel that it is going to have the immunity against the mutant strains?
I don't know, but that is one of the reasons why I said I would prefer to get a Covaxin. As a microbiologist, I feel it's the lysate, it's the mixture, it has everything.
Inactivated virus vaccine are always safer, and we have more experience with inactivated virus.
Inactivated viruses work very well. All polio, we've eradicated smallpox, we've worked with polio. We cannot attempt.
An unknown territory. No? It's an unknown word, and this is something.
I would like to ask a very, very basic question.
Dr. Gupta, this is your last question. People are raising their hands to me.
Fine, let them ask first, because this question does not belong to the clinical scenario.
No. I don't think they're just trying to.
I would like to know, what are the neutralizing antibodies? Is it possible to differentiate the binding the antibodies from the neutralizing antibodies, or it is just the virus neutralization or the plaque neutralization that we measure? Because the measurement facilities are there only with the research laboratories like NIV and all.
I agree. PRNT is not available everywhere. But neutralizing antibody is the antibody which is raised to the receptor binding domain of the spike protein, which neutralizes the virus. Now, this antibody is actually measured for plasma therapy. There are many kits now. Earlier, there was just one from Diasorin. Now, there are 2-3 companies which have made their CLIA-based test, which measure these neutralizing antibodies. And Diasorin, even in its product literature, has written that it correlates with the PRNT. So other companies also would have written. Similarly, there is a kit from China. I don't remember the name. That company also says that it measures the neutralizing antibodies. The rest of the kits, usually what we measure is an IgG or a combined antibody. Most of the CLIA-based tests measure combined antibody, not the IgG or IgM or IgA. So they just give you an antibody response, whether they are neutralizing or not, that you can know only with PRNT, or certain kits which have been approved after studies with PRNT. They've been correlated with PRNT.
Thank you, madam.
You're muted, Meenakshi.
Anybody else who wants to ask something or share the experience? So, if there are no questions, then I would like to thank Dr. Sonal Saxena for her excellent presentation. And I think the Z aspect was covered by Dr. Gupta. So thank you for the Z and thank you for A to Y. So with this, I end up today's meeting. And again, I want to pay my tribute to Dr. Vadhavan. So just remembering him, I want to end this session today. So thank you all for joining, and we'll meet again and we'll have another COVID update, vaccine update by Dr. Sonal. And I would like to give my special thanks to Dr. Amitabh for letting me know that you are such an excellent speaker. Thank you so much.
Thank you. Bye-bye.
Okay. Thank you, Dr. Shukla.